Yes, sir.
Yes, you can see the screen, right?
Yes.
Perfect. So in the last class, we finished chromosomal disorders, okay, non-Mendelian disorders. So there were just four disorders. So can I go today to Hiba? Hiba, are you there? Can you speak? Is your microphone working? Hiba, would you like to give me a brief recap of chromosomal disorders, the non-Mendelian disorders? No? Okay. Rifat, are you there? Would you like to give me a brief recap? Yes, Rifat, please. Thank you. Are you there, Rifat? Yes, I can hear something, but I cannot hear your voice. Can anyone hear Rifat? No? Hanifa, we come to you then.
Yes,
See,
sir.
people try. Yes, very good. Would you like to give me a brief recap of chromosomal disorders?
Yes.
Yeah.
Last class we started with sickle cell anemia, which is an autosomal and a recessive disorder.
Mhm.
H.
You're going back up, like you are talking about. Okay, would you like to start from sickle cell anemia? I am okay. Yeah, let's do this.
Sickle cell anemia is an autosomal and a recessive disorder. HbA and HbA indicates that the individual is normal, and HbA plus HbS indicates that it has the carrier for sickle cell anemia, and HbS and HbH indicates that the individual is affected with sickle cell anemia. Sickle cell anemia is caused by a single base substitution in the gene of the hemoglobin molecule, which is the beta chain, which leads to the substitution of glutamic acid by valine.
Yes.
Uh, and then we did we talked about mutations.
Yeah,
Point.
I just told you different type of mutations,
Yeah.
basically, that you should know. Yeah, point
The
mutation and frame shift.
mutation, and frame shift mutation. And then we went to phenylketonuria, which is also an autosomal and a recessive disorder. It is an inborn metabolic error where a single gene is affected. And then we talked about thalassemia, which is also an autosomal and a recessive disorder.
You forgot to tell how does it happen, remember? When there's a defect,
Yeah.
then phenylalanine is not converted to tyrosine.
When phenylalanine hydroxylase, it does not convert to phenylalanine, and which is not converted to tyrosine, then the individual is affected with phenylketonuria.
So phenylalanine starts rising up, and without phenylalanine hydroxylase, without this enzyme, the phenylalanine will automatically start converting into phenyl pyruvic acid. Now this is a dangerous thing, okay, harmful substance, and when it gets accumulated in the brain, it causes mental retardation. It affects the nervous system. If it gets accumulated in the skin tissues or the hair follicle tissues, they lead to loss of pigmentation in both. That's how it is also a pleiotropic gene, right?
Yes.
Yes. Now come to thalassemia.
Then we discussed about thalassemia, which is also an autosomal and a recessive disorder, which is due to a defect in mutation that reduces the rate of hemoglobin in both the alpha and beta chain synthesis. And thalassemia is different from sickle cell anemia. Like
Okay.
they're not the same thing. And then we did
How is it different?
Thalassemia is a quantitative thing, and whereas sickle cell is depends on quality.
Yeah.
And then we discussed about chromosomal disorders, which is the non-Mendelian disorders, which is one of them is Anemia, which is caused due to absence of abnormal arrangement of one or more chromosome in the genes, which causes
So
Down syndrome.
yes, so chromosomal disorders can be of two types. Okay, one is aneuploidy. Aneuploidy is gain or loss. It does not have abnormal arrangement. It is either absence or excess. Okay. Yes, continue. Now, Down syndrome.
Down syndrome, and then we have Turner syndrome, and then we have Klinefelter syndrome, and then we have.
So what is Down syndrome? It's trisomy of 21. Right?
Yes, the symptoms are mental retardation of furrowed tongue and partially open mouth, and physical or motor retardation. And for the Turner syndrome, it is the absence of X chromosome,
Yes.
which occurs only in females, and it's basically the underdevelopment of basic female characters. So the females with Turner syndrome are sterile, and their ovaries do not develop properly, and they lack the female secondary sexual characters. In Klinefelter syndrome, it is excess of X chromosome, which occurs only in males, and
Yes.
the men suffering from Klinefelter syndrome, the symptoms for them is they have an overall male body, but it causes gyno, gyno, gyno,
Gynecomastia.
gynecomastia,
Yes.
which is the development of breast in male, and they are sterile.
Yes. And we also discussed last one called Tau syndrome. Okay, this PA silent, it's called Tau syndrome, effectively. And Tau syndrome is trisomy, just like Down syndrome, it is a trisomy of chromosome 13. Down syndrome was a trisomy of chromosome number 21. Okay, and symptoms are similar: mental and intellectual retardation and physical abnormalities. Clear, everyone? No doubts?
Sir, last class you gave a question as homework.
What?
Sir, last class you gave a question as homework.
Yes, what was the question? Where is that question? Can you read the homework?
Yes, sir, 1 minute.
Yeah.
Sir, this was the question. Study the pedigree chart of a certain family given below and select
Okay.
the correct conclusion.
Great. So you did that. We did some questions, right? I realize.
Yes, sir.
Yes. Can you dictate me that pedigree quickly?
Sorry, sir.
Just dictate me the pedigree, like say a male and a female, and
Yes,
who
sir.
is affected. Yes. So tell me, what is the parent generation of that pedigree?
Yes, sir. An affected male is married to an affected female.
Yes, an affected male is married to an unaffected female, right?
Yes, sir.
And then what happens?
Yes, sir. There are 4 offsprings. The first one is an affected female.
Okay.
Second one is also an affected female.
Okay.
Third one is an unaffected male.
Okay.
And the fourth one is an affected male.
Affected male.
Yes, sir.
Okay. Good. Now, that's it.
Yes, sir.
Is there any offspring who got married to someone, or is there? So this is parent generation. This is F1. Is there F2 also in that question?
No, sir.
So
No. Okay. Yes. Tell me. Someone is saying something?
I said no, sir.
Okay, perfect. So let's start with this. So I hope Everyone solve this? So let me start with Atif because he reminded me of this. So Atif, what do you understand from this pedigree? Tell us. So what is it? It is autosomal or sex linked first. The second question is, I think this is what you have to find, right? Whether it is dominant or recessive.
No, sir. The options were the female parent is heterozygous, and then the second one was the parents could not have had a normal child.
Okay. So
Daughter.
basically, to answer that also, you have to go through this only. But tell me the options. First option is?
The female parent is heterozygous.
The female parent is heterozygous. Okay. Second?
The parents could not have had a normal daughter for this character.
Parents could not have had a normal daughter for this character. Okay.
The third option is the trait under study could not be color blindness.
The trait under study could be or could not be?
Could not be color blindness.
Not be color blindness. Okay.
And the last option is the male parent is homozygous dominant.
The male parent is homozygous dominant. Interesting options. Okay, so before going to these options, tell me what Atif, what do you think is the right answer? What have you understood?
Sir, one doubt.
Okay, you have a doubt to begin with. Good. Tell me, what is the doubt?
Sir, in the questions, like in the exams, will the carriers always be shown or not?
Carriers.
Yes, sir.
So,
Will they
yeah,
will they
sometimes
always be shown or not?
no. So
No?
sometimes what they do, it becomes easier because they color the half circle, right? And when you color
Yes, sir.
half circle, it means that those are the carriers, correct? This is what you are asking, that will this clue be given to you or not? Probably
Yes,
it
sir.
can be given to you in school exams or in board exams, but not in your entrance exams most of the times. If you get it in your entrance exams, then you are lucky, and then the question is easy. Tell me, what is your doubt here? Oh, this was the doubt only.
Yes.
So you tell me, what do you think is the answer according to you?
Sir, I tried the question by myself. I checked all of the options. I'm not very sure, but I think the answer is the first one.
First one. So this is what Atif thinks is the answer, right? Others?
Yes,
Hanifa, what do you think?
Yeah, I think it's the first one.
First one. Okay. So Hanifa also thinks it's the first one. Others?
So think it's the first one.
Fatima also thinks it's the first one. Who is it? Is it Fatima?
So
yeah. Fatima also thinks the first one. Others? Now everyone will think it's the first one. Rifat says it's A. She's so convinced. She's writing it's A. Yes. Done and dusted. Hiba, what do you think? Hiba is thinking, should I go for something? A. Okay. So every one of you thinks A, and I'm very, very happy because this is the right answer as far as I can also understand. So let's try to understand this pedigree. The second most closest answer is 4, but it's not making sense because this child is not affected. That's why. So let's go to figuring it out. See here, the male is actually affected, right? And the female is normal. Now, if the male is affected and the female is normal, and if girls in the next generation are also affected, then it has to be a sex-linked disorder. Do you understand?
Yes.
Because males always give X chromosome to the daughters. Yes? Do you understand?
Yes, sir.
Yes, sir.
Because at the worst, you have to apply this logic. And I think all of you applied this right logic that at the worst, what could happen is, but father can only give one X. And if that one X is enough to cause this disease, then it is a sex-linked dominant disorder. But but in this case, where is this child? This is a male, and why is he then affected? Because both the sexes being affected from just the father is not making sense. So sex linked is not working here, right? So clearly what's happening is, for it to be sex linked, this mother has to be a carrier, carrying one gene and one normal. And the father is actually this. So these two daughters happen to get XC from like this disease C's for that defect from the father, and also getting this X from the mother. Unfortunately, both are this, and this son only get this X and a normal Y from father, and that's how he is affected. You understand? And this son is not affected because it got a normal X from the mother and a normal Y from the father. Do you understand?
Yes, sir.
So among this, the male is a homozygous dominant. It can only be true for a autosomal disorder, right?
So,
Because a male can never be homozygous dominant for a sex linked. You understand that? For that matter, a male can never be dominant or recessive for X linked. It can either be a normal or an infected. So even if some disease is recessive, but it is on the X chromosome, it behaves like dominant in the males because males have only one X, right? Everyone?
yes, sir.
So this could have been the other closest. These two does not make sense at all. So these are wrong because the parents could not have had a normal daughter for this character. No, they could have had a normal daughter, but that normal daughter would be a carrier. Understand?
Yes, sir.
Not showing any traits. Now, the trait under study could not be color blindness. What is color blindness? Autosomal and what is color blindness?
Sex-linked
Quickly.
recessive disorder.
Yes, it's sex-linked and recessive. Okay. So you had to draw the right conclusion, right? Was the question asking the right conclusion or the wrong one? What was the question asking?
The correct.
The correct conclusion.
Correct one. Yes. So it will be A. Okay. Good. So everyone solved it good, and I'm very happy and proud that you understood this concept properly. Now, please go back, try to find more pedigrees wherever you can find them, any source, any pedigree, or there is a good exercise I used to do. Okay. So just tell anyone, just tell them that you have to make either a square or a circle, colored or non-colored. Ask your mother or your father to do this. Just scribble a chart for you, and then you should analyze it and see that if it is making sense, if it is a possible pedigree, not possible. If not possible, why it is not possible. Do you understand? Something should be just thrown at you, and this is a very easy thing. The rule is that only for parents there has to be one square and one circle, mother and father. After that, it could be anything, right? Yes or no?
Yes.
And if you have friends or like school friends whom you can meet, just make pedigrees and exchange. So this we used to do as, you know, our code decoding thing. So I used to think that I will make the hardest pedigree that no one will be able to analyze. Now, hardest does not mean it will be a 20 generation long pedigree. No, hardest means which I will think is hardest, and someone else will make a pedigree which is more convoluted. Sometimes that pedigree will be wrong, not possible according to logic, but then also you will learn that why it is not possible according to logic. So that you can do among yourselves. Okay, everyone?
Yes.
You all are connected through WhatsApp. So just scribble it on a page, click a picture, make a pedigree analysis group and say that, okay, today I'll send a pedigree. Whoever will solve it gets the chance to give the pedigree. You understand? Do it among yourselves. Okay, so let's start with the 6th chapter today. Can you see the screen? The molecular basis of inheritance. Now, whatever we have studied till now, you realize that genetics is very, very logical, right? Something is actually inheriting, and we know that it's gene, but now we are coming to molecular level. Now, what's happening molecularly? Not just the phenotype or how the disease is traveling in generations, but what's happening inside the cell on a molecular level. So to understand that, we have to understand all the players which participate in inheritance. So inheritance starts with gene, right? The concept of gene. Yes. And we know that gene is nothing but a physical fragment on a DNA only, right? It's a part of DNA. So let's first start with DNA. Let's know what is DNA then. What is DNA? It's a long polymer. We have done this. So just quickly keep answering so that we can move forward to the interesting things. What is DNA? Everyone. What does DNA stands for?
Deoxyribonucleic acid.
Yes, deoxyribonucleic acid. Okay, DNA. Now, what is it basically? It is a polymer. It is a long polymer. And if something is polymer, there has to have monomers of it. Polymer means a long chain of individual units, and monomer, the word mono means single, so that's single unit. Have you all seen a chain? In that chain there are individual small links that connect together. So one link will be this, the other will be this, and they will be linked like this, right? You understand? And then another one, another one, and it forms a chain. It's not like a rope continuous, it's like a chain. It has small monomers attached together to form a polymer. So it is a long polymer of deoxyribonucleotides, or commonly known as nucleotides. Okay, so nucleotides are the monomers, and when they join together they make polymer, which is called DNA. Is it clear, everyone?
Yes.
Now let's try to figure out the structure. What is a nucleotide? So in a nucleotide, so I'm making let's say there is a DNA. Okay, and from here onwards we start opening the DNA. Is it clear? So this one single chain, I should use another color for specific. Okay, so do you see this part of the DNA, this part? I'm going to make this part here. Okay, now let's see what happens in this part. So in this part, this is one single chain. So the backbone has phosphate group. Okay, so at the 5, this is called the 5' end, 5' phosphate. So at the 5' end, the DNA has two ends, 5' and 3'. Okay, so we are starting from 5' where there will be a phosphate, and then it will be connected to a carbon, which will be a part of a sugar. So that sugar will be a pentose sugar. What do we mean by pentose? 5 carbon. You see 1, 2, 3, 4, 5 structures, pentose sugar. And from that sugar, there will be a base coming out. Now this base can either be adenine, guanine, thymine, or cytosine. So there are 4 types of bases: A, T, G, and C, right? You all are familiar with this.
Yes.
We did this in the last chapter, we used it. A is adenine, thymine, guanine, and cytosine. These are the 4 nitrogenous bases in the DNA. So a 5' phosphate sugar attached to a carbon, there will be hydrogens here to complete the valency. This is ribose sugar, and this is a base, nitrogenous base. Okay, and this is
Yes.
one nucleotide as a unit. Now this keeps on repeating. So from here there will be another nucleotide attached. So sorry, let me make it better. So there will be another phosphate attached to carbon, which will be forming a phosphate sugar, and from that phosphate sugar will be attached a base. Now this time this base could be thymine, and then so on. You understand?
Yes, sir.
Another phosphate attached to carbon, which is attached to a phosphate sugar, and this sugar is attached to a base. Now this time it could be guanine. So ATGC, and then it continues. You understand? So this is a long chain, but if you want to know one link of it, like just the one monomer, this is the one monomer. Okay, everyone?
Yes.
This is nucleotide. Monomer for DNA. Please make it yourself, and you will realize that it's easy. So this sugar has to be five edges. 1, 2, 3, 4, 5. Attached to a carbon, phosphate, sugar, base. Phosphate, sugar, base. Okay. So this is the first thing, phosphate. This is the second thing, a sugar, and this is the third thing, a base. I'm writing it here. These 3 things together make the nucleotide. Third thing is base. Okay. Base can either be A, T, G, or C. Drawn, everyone? Now.
No, sir. No.
Yes, please draw this. And remember each part. Use a black here. So the phosphate is towards the 5' end, okay, always. And what is towards the 3' end? The sugar. Okay, so this side is the 5' end of this DNA, and that side is 3' end. So phosphate always is at the 5' end towards this side, and sugar is towards the 3' end. Okay. So this is true for this strand. For the other strand, it's opposite. 3' end and 5' end. Do you understand? The other strand which binds with DNA. So DNA is a double stranded structure. So if one strand, it's just like 2 lanes of a road. If in one lane the traffic is going from right to left, then in the other lane it has to go from left to right, right? 2-way traffic. Do you understand?
Yes, sir.
Yeah. Make this and then let me know. It's called a polynucleotide chain. And one single, which I have marked in red, is a single nucleotide. This.
Done, sir.
Done, everyone? Shall I move forward?
Yes, sir.
Now, if I ask you, so in this DNA, look here, these two are called the backbone of the DNA, right? So there are two backbones, 1 and 2, and in between it's just like a ladder. So ladder has two backbones on which the ladder stands, and then there are steps, right? So imagine DNA like a ladder which is twisted. So imagine a ladder, a straight ladder, and then you take the ladder and twist it to make it a helix. That's DNA. Okay. So the backbones, what do you think backbone is made up of? What makes the backbone? The phosphate and the sugar makes the backbone, right? And what, because this is a long chain that goes, right? This is the backbone. And what comes towards inside as a step? The bases. Bases protrude inside from that backbone. Do you understand, everyone? Is it clear? Atif, Anifa, Fatima?
Yes, sir.
Backbone. So write down phosphate and sugar. Phosphate and ribose sugar forms the backbone of the DNA. Forms the backbone of DNA polymer. And bases emerge from that backbone. Bases emerge from that backbone. Clear?
Yes.
Now, about the, write down certain points regarding the DNA. Write down. So this ribose sugar is 5 carbon, so it's also known as pentose sugar. Both are the same thing. Sometime in books you will read pentose sugar, which means the same thing. Okay. This is the name, this is the structure, and this tells you what the chemical is. It's a sugar. Now write down some characteristics of DNA. These question can be asked in boards. Write down what are, or give the characteristics of DNA. Molecular characteristics. The first thing, there are two types of nitrogenous bases. Types, by types mean the first type is called purines, and the second type is called pyrimidines. Purines are adenine and guanine, A and G. Adenine and guanine are purines. Then C and T, which is your cytosine and thymine, becomes your pyrimidine. Okay. Now remember that in case of RNA, thymine is not present. Okay. When a RNA is made from a DNA, remember, you know this DNA replication, transcription, you have read this. Do you know that from a DNA, RNA is made, and then from that RNA, protein is made? Yes,
Yes.
no, yeah. So in the RNA, there is A G, C, and instead of thymine, there is uracil, U. So uracil is a modified version of thymine only, which is present in RNA. So make a star here and write down it's present in RNA only. But it is a pyrimidine because thymine is a pyrimidine. Is this clear, everyone? The bases are of two types. Phosphate and sugar is common. They are the same. Bases are of two types. Okay. Now the rule is, write down rule of pairing. Bases pair to make those steps in the ladder. So the rule of pairing is purines bind with pyrimidine. And it's not like any purine can bind with any pyrimidine. No, it's specific. Adenine binds with thymine with a double hydrogen bond. So this here denotes double hydrogen bond. Is it clear? And guanine binds with cytosine with a triple hydrogen bond. Is it clear, everyone?
Yes.
And in chemistry, tell me, you have studied in 11th class, which bond is bigger? The length of single bond versus double bond versus triple bond. Which bond length is the biggest? Which is the longest bond? You've not studied this? Don't tell me. You must have studied bondings, right, Anifa? Which bond is longer?
The
Yes.
single,
Single bond is the longest, double bond is intermediate, and triple bond is shortest. So do you see the problem here that, you know, one bond is longer, one bond is shorter, but still we have to make two backbones exactly like railway tracks. So they have to be equidistant, right? That's why A bonds with T and G bonds with C, because they are not all of equal sizes. Okay. So this kind of pairing fits together and keeps the distance between the two backbones same throughout the DNA. Do you understand? Do you understand, everyone? Yes or no?
Sir, can you explain once more?
Yes, I'll try to draw it. Let's say these are the two backbones that you have to keep here only. And you have to join it. You have three things to join. This is a double bond. This is a triple bond. This is one big molecule. And this is one small molecule. Like this. And then there is another molecule. Like these are the ingredients I'm giving you. Okay. Let's say these are the ingredients. If you have to maintain the distance between them same, then at position 1, if you are already using a double bond, do you have space for these two molecules? Can we accommodate these two?
Yes.
So maybe you can accommodate the red one here. So it will take this space. Now, can you accommodate the black one also? No. So you have to go with the smaller green one because you cannot surpass this length. Do you understand this?
Yes.
Then if you take a smaller bond in the next, then you can use the black and also there is space, so you can use the blue, right?
Yes, sir.
Understand? So because this distance has to be maintained, that's why the sizes of ATGC are also different, and the sizes of the length of the double and triple bond is also different. So they are, that's why I said that A cannot bind with C, though A is a purine and C is a pyrimidine, right? A can only bind with T and G can only bind with C, because this thing only makes sense to keep the DNA equidistant, both the backbones equidistant. Now do you understand, everyone?
Yes.
Yeah. So in this case, let's say this was A and this was T. Okay. And then this was C and this was G. Okay. Clear? So this is the rule of
Yes,
pairing. One purine with one pyrimidine, which is A with T, G with C. Now write down the next point. Third point is, I'll draw it here only. Do you see this phosphate group binding with this sugar through this carbon? Yes.
Yes, sir.
This carbon is actually a part of the sugar. Okay. Part of the sugar. Now the phosphate binds with carbon through this bond. Now this bond here, write down the bond through which the phosphate group is linked to OH group of the fifth carbon. So this carbon is the fifth carbon. The bond with which the phosphate group is linked to the OH of the fifth carbon this bond is called phosphodiester bond. Sorry, phosphoester bond. Is it clear? Now you can see for a phosphate there are two such bonds, right? This one and this one. Both of them attached to sugar, right? One attached to the carbon and one attached to the direct sugar. You understand?
Yes.
Yeah, so then it becomes, if one is phosphoester, then the both will be together known as phosphodiester, because two. So phosphodiester bonding is between the phosphate and the sugar. Do you understand that?
Yes, sir.
So through phosphodiester bonding, you can keep joining as many sugars as you want with a phosphate as a linker. If you see it as a train, you know, so this is one bogie of the train, this is another bogie, and this in between is that linker which acts between the two bogies. You understand? Okay.
Yes.
Now about DNA, write down third point. DNA is a double helix structure. It's double stranded and it's helical. It's called. So it's called a double helix structure. And this double helix structure was proposed by, for the first time, it was proposed by James Watson and Francis Crick. Very, very famous scientists, because they figured out the DNA structure. Okay. And how did they figure out DNA structure? There were scientists who were trying to decode the structure, but they were not able to understand what could be the structure of the DNA. Now there were two scientists duo who was working on it. One was Morris Wilkins and Rosalind Franklin. You must have heard of the name Rosalind Franklin.
Yes, sir.
Yes, Rosalind Franklin, along with her apprentice Morris Wilkins, for the first time, they collected the data through X-ray crystallography. They did X-ray crystallography, also known as X-ray diffraction. So they did X-ray crystallography, collected the data, but could not understand it. So they should have been the actual persons who should have decoded it because they had the data, but they could not make sense of the data. They could not understand what kind of shape this data suggests. This data somehow reached James Watson and Francis Crick, who decoded the data. So all the hard work done by Rosalind Franklin and Morris Wilkins and James Watson and Francis Crick used their brains to figure out that this kind of data can only suggest one shape mathematically, and that shape is a double helix shape. Okay. So on the basis of their data, these two figured out the structure. Is it clear?
Yes,
Yes,
sir.
no? Clear. Okay, very good. So these are the characteristics of DNA. Now let's come to one more question. Write down this question. This is a very, very common question asked in boards. Write down. Explain the salient features. Explain the salient features, like the key crucial features of the double helix structure of DNA. It comes as a 3 marker or a 4 marker question in boards. Multiple times I've seen this question in boards. So write down the answer. First point. It has 2 polynucleotide chains. Okay, 2 polynucleotide chains where the backbone is made up of sugar and phosphate. Backbone of sugar and phosphate. And bases project inside. So always start with this point because this point just generalizes the overall structure. Okay. This point is clear, right? We have just discussed this. Then you have to talk about the polarity of the chain. Like I just said, one chain has 5' to 3' polarity, the other has an opposite polarity. So write down. Second point, the two chains have anti-parallel polarity. Anti-parallel polarity. Okay. So which means if one chain is 5' to 3' in this direction, then the other chain will be 3' to 5' in the same direction. You understand?
Yes, sir.
Now, third point. the bases from the two strands or chains, the bases from the two strands or chains are paired through hydrogen bond. Are paired through hydrogen bond. Okay, and then you can write adenine forms 2 bonds with thymine, and cytosine forms 3 bonds with guanine. Okay, so basically 1 purine and 1 pyrimidine. And it cannot be exchanged. It stays as it is. And why it stays as it is? Because I told you, because of that, to keep the width of those two strands constant. All right, done. You can also write it here. Just write a reason. This is to maintain the uniformity of distance between the two strands. Okay. And the fourth and the last salient feature is DNA is it's coiled in a right-handed fashion. Right now, coiled in a right-handed fashion. You know what does this mean? So in a right-handed manner, like this, from left to right, it's coiled. You understand?
Sir, clockwise?
Right-handed is, yes, clockwise. If you see it from the top, it's like clockwise towards the right.
Okay.
Okay. Okay. Now, write down, you know, when a DNA, let's make a DNA, okay? So from this point to this point, it's called a pitch of DNA. Is it clear? From one coiled
Yes.
region to another, it's called a pitch of the DNA. And write down, the pitch of a helix, the pitch of the DNA helix is exactly 3.4 nanometers. And it contains around 10 base pairs, 10 bases in each pitch or in each turn, because that turn is known as the pitch. You understand, everyone? This is a question which was asked in entrance. So if it is 3.4 nanometer, this distance, and it has 3 base pair in each turn, so we can figure this out, and we can figure that there is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 in each. So what will be the distance between two bases? Can you tell me roughly?
0.34 nanometers.
Yes, correct. So write down, distance between two base pairs is 0.34 nanometer. Okay, this is very difficult to find directly. So whenever something is difficult to find directly, if you know that in one matchbox, there is 100 matchsticks, and the volume of the total matchbox is, let's say, 200 centimeter cube, then you can tell me the volume of one matchstick, right? Just divide it by 100.
Yes.
Yeah. So this is the same thing done. Is this clear, everyone?
Yes, sir.
Okay. I think these are the features. These are most important characteristic features. Question is done. Now, DNA is clear to you? Any doubt in DNA? This is all you have to know about DNA. You have to know about packing of DNA, that I will tell later. But as a molecule, is DNA clear to you, everyone?
Yes.
There are other forms of DNA as well. So the DNA that we are studying here is called the bDNA. The B form of DNA. There is A, B, C, and Z. Okay. So just know it. It's not very important for your entrances, but just know there are many forms of DNA in living system. Normally, animals and humans, the most common, the normal form of DNA is this DNA. Okay. Cool. Now, having known about DNA, let me tell you about a concept in biology which is called central dogma of biology. It is called central dogma because it's followed throughout, and it's a very important concept. Now we all know that DNA can give rise to mRNA, right? Yes, everyone?
Yes, sir.
What is a mRNA? What is M here? M stands for messenger. So DNA makes a RNA. Why it happens is because, see, let me give you an analogy. You have a very important, a rare, single copy edition of a very important book, let's say, which you want to preserve and do not want to affect it. But all the laws, every rule that you will follow for something is written in that book. So you would like to keep, and that there's only one single copy of that book with you. So you would like to keep that book always safe, right? Because if something happens to that book, let's say oil or something spilled over that book, a page got torn or something, ink got smudged, then you will lose that information forever. There is no other copy from where you can just match it and get to know what did you miss. So in that case, whenever you require to know something from that book, will you take the book with you and travel? No, right?
No.
You will make whatever part is required, you will make a copy of it. Either you will take a photo on your mobile phone, or you will write down handwritten notes that are for reference, and you will move with that source of information, but not with the actual source of information, because you do not want the actual source to be exposed to any kind of damage or problem, right? That actual source in our body is DNA. DNA has information for each and every cells, each and every gene, each and every character of our body. If this gets damaged, then there is a problem. So what every cell does, it safeguards the DNA, keep it in a vault, in a locker, which is known as nucleus. And nucleus is a double layered locker. Nothing can go inside or outside the nucleus just like randomly, apart from some important molecules and factors and signaling molecules. So whenever you need an information out in the cell, in the cytoplasm, you do not ask the DNA to come out. It's like the king, right? Whenever a king decides something for the kingdom, does the king comes out of the palace all the time, stand on the street and declare what he has decided?
No.
Then what happens? A messenger of the king comes out, right?
Yes, sir.
And tells that this will be done. So let's say the cell needs your skin cell. Let's say you started partying on a beach. There is sun, there is water, and your skin now needs to get tanned to protect you from the UV radiation of the sun. So you need more and more melanin, and that melanin is a protein, the gene for which is in the DNA. So the cell needs, your skin cell needs more melanin. So DNA will just make the copy of that gene for melanin in the form of a mRNA, a messenger RNA, which will go out in the cytoplasm, and this RNA will be read by ribosomes. You remember ribosomes?
Yes, sir.
What did ribosomes used to do in the cell? What are they called commonly? Protein synthesizing factories of the cell. So ribosomes produce proteins from this mRNA. So DNA makes mRNA, mRNA gives rise to protein. Is it clear?
Yes, sir.
DNA can replicate to make more DNA itself. This is called replication, genome replication, which happens before every cell division. You know that?
Yes, sir.
How does a 2n cell divides and still remains 2n and 2n? Mitosis. It had 2n of information. Now, if you together add, this is 4n of information, right? Because before division, this 2n replicated and made 2n plus 2n. Is it clear? That's called DNA replication. But it only happens before cell division, and this process is known as replication, DNA replication. The process by which DNA makes mRNA is called transcription. Make sense? We are making a transcript from the original source.
Yes.
You know the meaning of a transcript, right? And then this source also has the same message. So now this message will be translated. So this is called translation. Replication, transcription, translation. Is it clear?
Yes, sir.
Some viruses can actually tweak this central dogma. They are an exception to this central dogma. Okay. We normally make DNA from, we normally make mRNA from DNA, but some viruses known as the retroviruses, they can actually reverse this flow. They can actually make a DNA from an mRNA. This is a very special and unique case for viruses. So what will this called? If you are able to make DNA from an mRNA, if the forward process is called transcription, what will be the reverse process called? How many of you drive a car or a bike? There is a gear. To go back, there is a gear. What is to go back?
Reverse.
Sorry, come again, Atif, your voice broke.
Reverse.
Reverse gear, right? So if there is a transcription, what will be the opposite?
Reverse transcription?
Yes. Is it difficult to remember?
No.
So reverse transcription can be done by viruses. Okay. So they create an exception, or they create a backward flow in the central dogma of biology. Is it clear, everyone? Also write down. Also write down. So at this step, from mRNA to protein, ribosomes form, write down, ribosomes read mRNA in the cytoplasm and form proteins. Ribosomes read mRNA in the cytoplasm and form protein. Is the central dogma clear to you? So what does this central dogma states? Write down. It states that the genetic information flows from DNA to RNA and RNA to protein in every living cell. And the exceptions are viruses that can actually do reverse transcription. They can reverse the flow. Is this clear, everyone? Shall I move forward? Done?
Yes.
Okay. Now you have understood DNA. You have understood that RNA is formed from DNA and then protein is formed, and that's called central dogma, the flow of genetic information. You have understood the characteristic property of the structure of DNA. Now, as I told you, DNA is a long polymer. It's like a very long thread. Now, do you know that if you take all the DNA in your cell, okay, all the DNA means how many chromosomes do we have in the cell?
46.
46. If we take all the 46 chromosomes, what are chromosomes basically? That you also know. They are, last chapter we talked about, they are super coiled form of DNA, right? I gave you the
Yes, sir.
example of a thread bound over a spool, right? A spool of thread. So DNA is a very long thread bound over the spool. That's why we can keep it in a very small space. If you unwind all the DNA, all the chromosomes, in one single cell, the length of your DNA will be more than 2 meters. If you unwind all that 46 chromosomes and place it end to end, then those 46 chromosomes will span 2.2 meters, more than the length, more than your own height. And that is the amount of DNA present in one cell. And how many cells do we have in the body? Trillions of cells, trillions. You know, we have around 90 billion just in our brain neurons. That is just 1 kg organ, 90 billion neurons. And we are talking about trillions of cells everywhere in our body, and every cell has a diploid set of genome, 46 chromosome. So if you take just one person's total DNA, like any one of us, like if you take my total DNA from each and every cell, place it end to end, can you imagine where can it go to? What will be the length of that total DNA? Any guess? Where can it go to from? Yeah, tell me. Yes, Aditya. Any wild guess? Wildest guess you can make.
Sir, maybe like it like reach astronomical lengths, maybe like go around the world 3
Yes.
times or something.
Not just the Earth, it will span the whole solar system. Trillions of cells, 2 meter into trillions of meters becomes billions of kilometers. From Earth to Saturn is around, 600 billion kilometers. So 6 billion kilometers, something like that, if I'm not wrong. So we can understand what lengths we are talking about of the DNA. And we can also understand what type of packaging we are talking about here. So that 2.2 meters, write down. The next topic that we are going to discuss, it's very interesting how biology has found this way. It's miraculously magnificent that we are now going to study the packaging of DNA. in a cell. Okay. So write down how do we basically calculate this whole length of DNA? So if you calculate the total length of DNA, what I was saying, it's not in like thin air of DNA in one cell. Let's start with this. So tell me how much did you tell me was the distance between two base pairs?
0.34 nanometers.
Yes. So first thing, let's take the shortest. The distance between two base pairs is equal to 0.34 nanometers. Now you know how many base pairs does a now if it is in nanometer, and if I want to convert it into 10 raised to power something, if you take all that decimal in meter, what will be the meter? 0.34 nanometer is
0.34 into 10 raised to minus 9 meter.
yes, 0.34 into 10 raised to power minus 9 meter, right? Nanometers is raised to power 9. Now second thing, how many total base pairs are there? Any idea? Total number of base pair? It is 6.6. I'm taking the double diploid set of the genome. If it is haploid, it is 3.3. So a gamete has 3.3, but a normal cell has 6.6 into 10 raised to power 9 base pair. Okay. Now if you multiply this with this distance, which is 0.34 into 10 raised to power 9 9 meters, what will be the total distance? Can you solve this mathematical expression? You have to multiply this with this. 10 raised to power 9 will just cancel out, right?
Yes, sir.
You're left with 6.6 into 0.34. How much is that?
Sir, 2.2 something.
Yes, 2.2. And what was the unit here? Meter. So 2.2 meter is the total length of the DNA in one cell. Okay. Do you realize how we calculated it?
Yes, sir.
Isn't it mind-boggling? We cannot see a cell with the naked eye, and it has a thread which is 2.2 meter long. Of course, it must be very, very thin, not visible. One thread is not visible to the naked eye, but it is a long polymer, very long polymer. Yes or no? And this 2.2 meter thread has 2 copies of our genome, one from the father, one from the mother. And it contains all the genes that we are made up of, all the characters that are present in our body. Okay?
Yes, sir.
Yeah. So this is one cell. Now, when we come to packaging, how do you think it must be packaged then? Remember, I told you very early in reproductive, when we were discussing some concepts, that to wind the DNA, we need something, and that something is protein, right? Just like to wind a spool, a thread over a spool, there has to be a cardboard frame on which we wind the thread, right? So that thing is called histone protein. So DNA, write down, DNA are bound or wound around histone proteins. Okay. You know, DNA is negatively charged, everyone. DNA is a negatively charged protein because of the phosphate group. It's a negatively charged molecule or a negatively charged polymer. So if it is a negatively charged polymer, then what should be the nature of the protein on which it has to be bound tightly? Positive, right? So histone proteins are positively charged. Is it clear? Do you understand? Is it clear? Am I audible? Atif, Rifat, Fatima, Hiba. Now that, am I audible or no?
Yes, sir.
Yes. Okay. Now this binding has to be very, very tight, and it has to be on multiple levels. Just by binding it will not help. So one thing that happens is there's a very long, let's say this is a long thread of DNA. Now, after every some time, you introduced a protein. Okay, let's say this was negative charge, this is positive charge. So what will happen? The structure will look something like this. After every some time, there'll be a chunk of it bound, right? Yes. And the whole 1, 2, 3, 4, 5, 6, the whole length of the DNA will be smaller now. You understand? Is it clear, everyone? So this length becomes smaller by binding. Is it clear? Now let me draw one of this in detail. So I will use, I'm drawing this part here. So there is a big molecule. It's actually not one, it is octamer. Okay, these are 8 protein molecules together, which looks like a big spherical molecule protein. Now this is called histone octamer because there are 8, okay, or core histones. Now over this, does anyone has a question? Okay, Atif, it's okay. Now I'm using black as the DNA. Now this DNA binds like this. So it comes from one direction, goes, makes a circle around it, comes from another direction, goes, and then comes out. So you see that the DNA is wrapping around the histone core 1, 2.5 times. So if you include this and this part, it will be 2.5, right? 1, 2.5. Is it making sense? And this part is then kind of stapled with another molecule, which is a long molecule. Now this is known as H1 histone. So all are histones only. This is H1 histone. In the black, this is the DNA. And this blue is a histone octamer, 8 subunits of histones together, histone core or histone octamer. And this is H1 histone. Is this structure clear to you, everyone? Everyone, is this clear to you? At least say something or write something in the chat. This is very important diagram. It comes in boards. Draw the diagram of a nucleosome. So this is what I'm trying to tell, that this part is called, this is the first level of packaging. And this is called a nucleosome. What it is called? A nucleosome, where DNA goes 2.5 times around a histone octamer and is stapled by H1 histones. And when these structures look like this type, when you look in the cell, there are many histones with DNA wound around it. Then this kind of structure is called beads on the string. Beads on string structure. Is it clear? So do you understand now what is beads on string and what is a nucleosome? Everyone? Fatima, Heba, Rifat? Yes, please draw this and let me know when it is done. Question is, with the help of a diagram, define the structure of a nucleosome. And this kind of structure happens in a eukaryote. In a eukaryotic cell. Draw this first. Draw this bead on string structure also. Let me know when you're done.
Done.
Done? Okay. Just give me one moment. I'm trying to pull out diagram if I can get it. Still. Yes. This is a better diagram. Am I still audible and visible to all of you?
Sorry, sir.
Am I audible and visible to all of you?
Yes, sir.
Okay. Now let me try to. Yes, it's here. People, let's try to understand it with this diagram. Can you all see this diagram that I've just pasted?
Yes, sir.
Let me try to keep it in. So this is the copyright from Benjamin Cummings. Okay, it's hope that describes this. You all can see the diagram, right?
Yes, sir.
Yeah. Now pay attention here. We use blue. So this is the double helix. Can you see this? The distance between the two strands is 2 nanometer. The distance between the two bases is 0.34 nanometer. Yes. And the distance between this one pitch is 3.4 nanometer. Yes, everyone?
Yes, sir.
Then this DNA, if it has to first layer of coiling is what we just studied. It makes nucleosomes. So you see these histone proteins, these histone proteins, round one. So they wrap around these histone proteins like this. So here you can see it's an octamer, 1, 2, 3, 4, 5, 6, 7, 8. So I have just shown it as one big gola here. Actually, this gola is 8 different small golas, okay, called a octamer, histone octamer. So these histone octamers, can you see nucleosome now? They call it a nucleosome here.
Yes.
So this is exactly what we studied here. Okay, this is the nucleosome. On an octamer, there is a DNA wound. Okay, so this is first layer of coiling. And when these octamers line one like one after the other, they form a thread. So this also is looking like a thread to you, right? This, which is called a chromatin. Okay. And then this chromatin again starts supercoiling. So that's called supercoiled structure. And this supercoiled structure finally will make a chromosome. Do you understand? So at the very end, it will be a chromosome. So do you understand there are many layers of supercoiling happening? The DNA is coiling around the histones first, then the histones are arranging, like folding with each other. And then that whole thread of chromatin is also undergoing supercoiling. Do you understand? And after 3, 4, 5 layers of supercoiling, we come to the end product, which we see as chromosome. So this one thing that I showed you in the last picture, can you now imagine that how much DNA is present here, like supercoiled form?
Yes.
Yes, it's called this chromosome. Okay, is this clear, everyone?
Yes.
So now start writing down some things. Write down the positively charged basic proteins are called histones. Positively charged basic proteins called histones around which the negatively charged DNA is wrapped. Around which the negatively charged DNA is wrapped. To form a structure. To form a structure called nucleosome that you just made here. This one. to form a structure called nucleosome. Okay. Now, a typical nucleosome, this one, a typical nucleosome, one nucleosome contains 200 base pairs of DNA. So from this point to 1 round, 2 round, and coming back here, how many bases are in between? How many base pair are there? 200, which means how many pitch, how many turns are there? 200 base pairs are there. In one pitch, there are 10 base pairs, right? So 20 turns will be there, right? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Such 20 turns will be around one nucleosome. 20 turns means 3.4 into 20. That much nanometers of the DNA. You understand?
Yes.
Around 64 nanometer of the DNA is in one nucleosome. So you can do all this calculation if I just tell you that 200 base pairs of DNA helix is in the nucleosome, right? You can do all the calculation going back. Yes. Now write down from there onwards. Nucleosome makes the repeating unit of a structure. So see this unit 1, 2, 3. So they are repeating now, right? So nucleosomes make repeating units of the structure in the nucleus and is called chromatin. Chromatin. So this is called chromatin. Second level of supercoiling is chromatin. And write down chromatin is thread-like stained bodies. So if you look at a chromatin, you will see this body as one, this body as one, right? You will see something like this, right? Does it look like body after body? There are something, a bead-like thing.
Yes.
So they form thread-like stained. You can stain it with any DNA staining color. Thread-like stained bodies seen in the nucleus. Okay, and this is called nucleosome and chromatin both. So the nucleosomes in the chromatin is known as, write down, bead on a string, where the bead is the nucleosome. Can you see this individual bead? 1, 2, 3, 4, 5 I have marked. And this is chromatin. So bead on a nucleosome. Sorry, bead on a string, where bead is the nucleosome and chromatin looks like that string. Okay, clear till here? So again, let's begin. It's the naked DNA. So this DNA is called naked DNA, not bound on anything, not wrapped around anything. Naked DNA wraps around histone proteins, the histone octamer, and forms the first level of coiling known as nucleosome. In a nucleosome, around 200 base pair of DNA is there per nucleosome. These nucleosomes start coming together and form a bead on string like structure known as chromatin, second level of coiling. This chromatin then supercoils, write down, to form. The chromatin is further coiled. The chromatin is further coiled to form chromatin fibers. So do you see this chromatin now going and making these fibers as a? So this is one fiber. Can you see this, everyone?
Yes.
So this is called one chromatin fiber. Chromatin fiber has chromatin supercoiled in it. So chromatin fiber is the third level of supercoiling. First is nucleosome, second is chromatin, which is known as bead on string structure. Third is chromatin fibers. Now write down. Chromatin fibers are further condensed. Chromatin fibers are further condensed at the metaphase stage. Remember, in the metaphase stage, everything aligns on the plate, right? Like this. It is called metaphase plate. The DNA aligns like this. Then half of it goes to one side and half of it goes to another side. You have seen this diagram, right? In cell division. And then cell divides from the center. Yes. So this thing which aligns at the metaphase during cell division is these chromatin fibers which align to form chromosomes. So write down, chromatin fibers are further condensed at metaphase stage, condensed at metaphase stage of cell division to form chromosomes. So can you see here also I have shown these are the chromatin fibers. They will keep on condensing to give rise to this final structure called chromosome. Is it clear? So let's go to the sequence again. This can be asked. Sequence of DNA supercoiling. That's why it's called supercoiling, because coiling is not just happening once. So first, the naked DNA forms what? Nucleosome, right? Then these nucleosomes form chromatin. At this stage, it is called bead on string. BOS I'm writing. Bead on string structure. Then this chromatin condenses to form chromatin fibers. Yes. And this chromatin fiber condense at the metaphase to form chromosomes. So at 4 levels, starting from naked DNA, there are 4 levels at which coiling happens. That's why it's called supercoiling of the DNA. Is it clear, everyone?
Yes.
Pakka? Just go through everything once. And let me know if there is any doubt. If everything is clear, then we are good to go forward and start with some experiments, molecular experiments. Go through everything from the beginning. Starting with the structure of DNA, we realize what is DNA, characteristic of it, base pairing, then how was the structure found, who found the structure, salient features, then central dogma. Then we started with packaging of DNA. We first found out how much DNA is present in one cell. Then DNA is negatively charged. Then there are proteins which are positively charged. And therefore, in a eukaryotic cell, the DNA is wound around that protein to form a bead on string structure, which is called a nucleosome, which looks like a bead. And then we went on to this figure to understand what's happening. These are the levels of supercoiling to finally from a DNA to form a chromosome. Is it clear? Everything is clear?
Yes.
Okay. Now, in the chromosome, there are certain regions of the chromatin. Okay. Some regions of the chromatin are loosely packed together. Some are very tightly packed together. Now, if something is loosely packed together and you put color to stain it, then you will see that that region, because it is loosely packed, the stain will move through it, right? And it will be lightly stained. But the region which is tightly packed, stain cannot move through it, and it will hold the stain, and those regions will be tightly packed. Dark stained. Do you understand?
Yes.
So on a chromosome, if I make a chromosome, let's say, okay, if you stain a chromosome, you will see that some regions are stained very dark versus the other ones are stained very light. Do you see this difference in staining? Then again, some dark regions will be there, and some light regions. So what is the dark region telling you about? Dark stained region. This is telling you that the chromatin is tightly packed, very, very super coiled. You understand?
Yes.
And what is this light region telling you? Is it visible what I'm writing with the light stained region? I hope it is visible.
Yes.
Is it visible, everyone? Yeah. So this tells that the chromatin in this region of the chromosome is loosely packed. Okay. Now, if a region is very tightly packed, okay, then do you think that that region will be accessible by the proteins which make RNA from the DNA? Transcription will be easy in which region? Suppose, let me give you an analogy. You are moving through forest, okay, Atif?
Yes, sir.
Then there comes one patch of the forest where there are trees, but they are not very close together. There is space between the trees, and you can easily move through that patch. Then you enter a patch where there are bamboo plantation, bamboo trees very close to each other. So bamboo trees, when they grow very close to each other, they are like they form a barrier. It's difficult to move through them because they do not have space in between. They are tightly packed, and they are straight trees. So in which patch of the forest you will be easy going? You will feel more accessible.
The trees,
Tightly
space
packed,
between them.
loosely packed, right?
Yes.
So for making RNA from the DNA, there are also molecules that have to reach the DNA, open the DNA, then make an RNA from one strand, and then close the DNA. Correct?
Yes.
DNA is double helix like this. First, a molecule has to reach that DNA, open it from one strand, make a RNA, then close it and take that RNA out of the nucleus, because that is now will be used for making protein in the cytoplasm. Do you understand, everyone?
Yes.
So in which region of the chromosome do you think transcription is easy? In the light stained region or in the dark stained region?
Light stain.
Yes, everyone understands the concept. In the loosely packed region, transcription will be more. Yes, everyone? Say
Yes,
yes or no? Yes, I know you understood. Others? No one is speaking. Okay. So I assume that you understood. So this dark stained region is known as heterochromatin. What is it called? Heterochromatin. and heterochromatin is transcriptionally inactive region. You understand why?
Yes, sir.
Molecules cannot reach the DNA, they cannot open the DNA easily, and they cannot make RNA out of it. But this light stained region is called euchromatin. This is basically the true chromatin. U means true, and euchromatin is transcriptionally active. So transcription is happening here. Okay.
Yes, sir.
And this happens because of the supercoiling. Now you tell me, now I ask you a question. Every cell of our body has the same genome, right? Yes? Yes or no?
Can you repeat the question?
Every cell of our body has the same genetic material, same number of genes, same genome, everything is same. Am I audible?
Yes, sir.
Was my question audible?
Yes, sir. It was audible.
So what is your answer?
No, sir. I don't think so.
So you think that all the different cells in our body have different genome, different genes?
Yes, sir.
No, our whole body is formed from a single zygote. So whatever was in that zygote just got copied and pasted in every cell. Yes or no? The same 46 chromosomes were there in the zygote. 23 came from the mother, 23 came from the father. The same 46 chromosome is in the cell of the eye, the cell of the brain, the cell of the bone, the cell of the skin, the cell of the blood vessel, the cell of the uh, gonads, the cell of the heart, the cell of the liver, kidney, every organ. Same 46 chromosomes are there in our body. Yes or no?
Yes, sir.
Yes. Then how and why are all our cells different? Some become circulatory system, some become nervous system, some has different function, some has different function. Do you understand the question? How from one single ancestor, which is a zygote, when the embryo develops, there are so many different kind of organs and cells formed. Can the muscle cell do the same function that a neuron cell do?
No.
So they have very different functions, very different structures. Can a fat cell do the same function that a liver cell does? No. But all these cells have the same genome sitting in their nucleus. So how do they become different? Think about this question, and I'll see you in the next class where we will try to find out the answers for this question. Okay? Because if you understand this, you are ready for entrances. Okay, everyone?
Yes.
Think about this question. Take this question home. Everyone understood what I asked? How different types of functional and structural cells are produced from one single zygote having the same genome is the question. Think about it. Search it, try to understand it. We'll continue the discussion in the next class. Okay, everyone? Any doubts? Let me give you a hint. The hint lies here in euchromatin and heterochromatin. But okay, we'll continue the discussion in the next class. So bye-bye, everyone. Good night. Take care.
Thank you.
Bye-bye.
Thank you, sir.
Bye-bye. Welcome.